I have nothing to declare. Hi, my name is Stephanie Tremblay from Concordia University. I'm going to present my work on multivariate quantification of brain differences in individuals who are genetically at risk of developing Alzheimer's disease. As there is currently no cure for dementia, it is crucial to detect the disease as early as possible. Alzheimer's disease is thought to result from a complex cascade of events that can vary from individual to individual. So it is a very heterogeneous disorder, and the pathological mechanisms are still debated. Recent studies have reported white matter alterations as one of the earliest changes in the disease time course. Studying individuals with a family history of AD and with the ApoE4 genotype provides a unique window to investigate brain abnormalities in this early stage. To detect these subtle early changes, it is crucial to use highly sensitive methods. One way to do that is to use multivariate approaches. Our objective was to assess differences in white matter microstructure in individuals that are genetically at risk of developing Alzheimer's disease using a multivariate framework that was developed by our group. The MRI data of 124 cognitively unimpaired older adults with a family history of AD from the Prevent AD dataset were analyzed in this study. 49 of these subjects had at least one E4 allele of the ApoE gene. They will be referred to as the ApoE4 group. 75 subjects had the A3 allele and will be referred to as the control group. I fitted the DTI and NAUTI model to the DWI data and performed constrained spherical deconvolution to obtain pixel metrics. I also used the T1 map from the MP2 rich sequence and calculated the T1 over T2 ratio. 12 types of maps were thus generated for each subject. I then computed group average from the subjects of the control group, which served as our reference group. And I computed the covariance matrix between all features from the subject of the reference group. This all served to compute what is called the Mahalanobis distance, or MADI, between each individual of the ApoE4 group and the reference group average. MADI is a multivariate distance measure between a point and a distribution. With this method, covariance between features is accounted for, and variables are scaled by dividing by the covariance matrix. This transforms the data into uncorrelated data. This computation was repeated at each voxel included in our masks of analysis. I did two separate analyses, one in a white matter mask and one in the subcortex, because we expect the metrics to have different meanings in white matter than in subcortex, and this was confirmed by the different correlations seen between features in the two tissue types. Here we can see the mean multivariate distance map of the ApoE4 group. The bright yellow areas are the brain regions that differ the most from the reference group. We can also see the histogram of the distance in all voxels. To investigate whether the differences seen are due to the presence of the genotype and not just to inter-individual variability, I then computed MADI between each individual of the control group relative to the same reference group. As we can see from the histogram and the maps, the distance values are much smaller. To confirm this, I then conducted a two samples t-test on the MADI maps. MADI was significantly different between the two groups in the genu of the corpus callosum and in white matter tracts near the angular gyrus, which is a region involved in several cognitive processes. Similarly, in the subcortex, the MADI distributions are very different between the two groups, with overall higher distances in the ApoE4 group. MADI was significantly different between the two groups in the hippocampus. In this study, we identified several regions where MADI was significantly greater in the ApoE4 group than in individuals without the genotype. This suggests the presence of white matter microstructural abnormalities in several major white matter tracts in these cognitively normal individuals with a genetic risk of developing AD. This included tracts projecting to the angular gyrus and hippocampus, both regions known to be affected in AD patients and that are associated with impairments in several cognitive domains. In future work, the link between MADI and cognition will be investigated to assess the clinical relevance of these differences. I will also extract the metrics that contributed most to the multivariate distance in these regions to reveal the potential biological mechanisms that underlie these differences. For more details on this, I invite you to attend my oral session. Thank you for your attention.